The purine and the pyrimidine bases, as you see, stick out to one side of a sugar phosphate backbone. In each DNA molecule, the number of nucleotides containing adenine, A, equals the number of containing thymine, and the number containing guanine, G, equal the number containing cytosine, C. As you see, adenine base pair with thymine by 2 hydrogen bonds, but cytosine base with guanine by 3 hydrogen bonds. The direct evidence for the structure of DNA comes from X-ray diffraction pictures made by passing X-rays through crystals of molecules having a regular structure. So now we are going to talk about how can we recognize this structure. It comes from X-ray diffraction pictures that made by passing X-rays through crystals of molecules having a regular structure. These produce a pattern of dots, give information about shape of molecule. In 1952, Franklin produced such photographs for crystals of highly purified DNA. Her results showed that, first, DNA is twisted into a spiral or helix. Second, it was bases perpendicular to the length of the fiber. Third, sugar phosphate backbone is on the outside of the helix with the bases on the inside. Fourth, the diameter of the helix showed that it must be composed of more than one strand of DNA. I am going to talk about the Franklin photographs and its result. So it resulted 4 things. First, DNA is twisted into a spiral or helix. Second, with the bases perpendicular to the length of the fiber. Also, sugar phosphate backbone is on the outside of the helix with the bases on the inside. The diameter of the helix showed that it must be composed of more than one strand of DNA. Franklin made her picture arrays, started to put all the available data together into a consistent model of DNA structure. After efforts of Franklin, we have efforts of British scientists Watson and Crick were the first to put an acceptable model for DNA. They said that the model of DNA structure put by them consists of 2 strands of DNA. The 2 strands are arranged like a ladder, with the ladder sides being the sugar phosphate backbones of the 2 strands, and the rungs being the bases. A rung may consist of either an adenine paired to a thymine or a guanine paired to a cytosine. In each rung, either base may be on either strand. The pair of bases in each rung is held together by hydrogen bonds. Hold an adenine thymine pair together by 2 hydrogen bonds, and guanine cytosine pair together by 3 hydrogen bonds. Each pair consists of 1 single and 1 double ring. Also, all the rungs of the ladder are of the same width. The backbones of the DNA strands are always the same distance from one another. 4 hydrogen bonds to form property between the base pairs in DNA, the 2 nucleotide strands had to run in the opposite directions, with the 5' free 5' phosphate group of the two strands at opposite ends of the molecule. Although the whole ladder of the DNA is twisted with 10 nucleotide pairs per turn, I mean 10 nucleotide pairs per turn, to form the spiral detected by Franklin's X-rays photographs. Because spiral is composed of two strands wound around each other, the DNA molecule is referred to a double helix. I am going to talk about the structure of DNA from view of Watson and Crick again. So, the efforts of Watson and Crick are summarized in that the model of DNA structure put by them consists of two strands of DNA. The two strands are arranged like a ladder, with a ladder side being the sugar phosphate backbones of the two strands and the rungs being the bases. The rung may consist of either an adenine paired to a thymine or guanine paired to a cytosine. In each rung, either base may be on either strand. The pair of bases in each rung is held together by hydrogen bonds. Hold an adenine-thymine pair together by two hydrogen bonds, and guanine-cytosine pair together by 3 hydrogen bonds. Each pair consists of one single and one double ring. All the rungs of the ladder are of the same width. The backbones of the DNA strands are always the same distance from one another. 4 hydrogen bonds to form property between the base pairs in DNA, the two nucleotide strands had to run in opposite directions with the free 5' phosphate groups of the two strands at opposite ends of the molecule. The whole ladder of DNA is twisted with 10 nucleotide pairs per turn. I mean 10 nucleotide pairs per turn to form the spiral detected by Franklin's X-ray photographs. Because the spiral is composed of two strands wound around each other, the DNA molecule is referred to double helix. Now, let's answer some question about this part of the. Choose from the achievement in column B what is suitable for the scientist name in column A. First, Hershey and Chase. Second, Watson and Crick. 3, Griffith. 4, Avery. 5, Franklin. In column B, gave us the acceptable model of DNA molecule, describe the bacterial transformation, notice that the amount of DNA is twice in some cells, provided that DNA is the genetic material of bacteriophage, describe the structure of DNA by using X-rays diffraction, isolated the material that inducing genetic transformation in non-virulent bacteria. I am going to say what in column B again. Give us the acceptable model of DNA molecule, describe the bacterial transformation, notice the amount of DNA is twice in somatic cells, provided that DNA is the genetic material of bacteriophage, describe the structure of DNA by using X-rays diffraction, isolated the material that inducing genetic transformation in non-virulent bacteria. So, first one, Hershey and Chase. What are you going to choose? Great, provided that DNA is the genetic material of bacteriophage. Next, Watson and Crick. Good, give us acceptable model of DNA molecule. Griffith, describe the bacterial transformation. Avery, excellent, isolated the materials that inducing genetic transformation in non-virulent bacteria. Franklin, at last, you are right, describe the structure of DNA by using X-rays diffraction. Let's move to another item of question. Choose the correct answer. The enzyme which need in building DNA is polymerase, pepsin, helicase, deoxyribonuclease. You are right, the enzyme which need in building DNA is polymerase. Next, deoxyribonuclease enzyme hydrolysis. Protein and DNA, RNA, proteins, RNA and proteins, DNA, carbohydrates. Let's think together. Deoxyribonuclease enzyme hydrolyze what? You are right. Deoxyribonuclease enzyme hydrolyze DNA. Which genetic material contains space? Sulfur, RNA, phosphorus, phosphate, ribose, sugar, or ammonia? You are right. Which genetic material contains phosphorus? Next, for a DNA double helix, which of the following statements is correct? Adenine base pair with guanine by 2 hydrogen bond, cytosine base pair with thymine with 2 hydrogen bond. I mean adenine equal to guanine, cytosine equal to thymine, adenine equal to cytosine, guanine equal to thymine, or adenine plus thymine equal to guanine plus cytosine, adenine equal to thymine, and guanine equal to cytosine. Of course, the answer is going to be for DNA double helix, the statement which is correct, adenine equal to thymine and guanine equal to cytosine. And you know adenine base pair with thymine by 2 hydrogen bond, but cytosine with guanine with 3 hydrogen bonds. Next, the biological information molecules are space, proteins, genes, chromosome, nucleic acids. Of course, the biological information molecules are nucleic acids. Next, which of the following nucleotide parts bond with each other with covalent bonds to form the DNA backbone? The sugar and the phosphate, the sugar and the nitrogen base, the phosphate and nitrogen base, none of all. Of course, the answer is going to be sugar and phosphate. For a DNA double helix, which of the following is not correct? Guanine plus adenine equal cytosine plus thymine, adenine equal to thymine and guanine equal to cytosine, adenine plus thymine equal to cytosine plus guanine, adenine plus guanine divided thymine plus cytosine equal 1. Yes, the answer is going to be adenine plus thymine equal to cytosine plus guanine. Next, the barriers of genetic information is space, proteins, fatty acids, RNA, or chromosomes. Of course, the barriers of genetic information is chromosomes. A crucial experiment which explained that the DNA is the genetic material is pneumonia bacteria, is vagus bacterial transformation, deoxyribonuclease enzyme, or ligase enzyme. The answer is going to be the crucial experiment which explained that the DNA in the genetic material in pneumonia bacteria is deoxyribonuclease enzyme. Let's move to another item of question. Give reason for. The mice of experiment infected by pneumonia then died after injection by a certain kind of bacteria, but in another case, after injection, infected by the same disease by another strain, but not die. Again, I'm going to say this question again. The mice of the experiment infected by pneumonia then died after the injection by a certain kind of bacteria, but in another case, after injection, infected by the same disease by another strain but not die. Of course, the answer is going to be because there are two strains of bacteria. First, strain called virulent bacteria S causes pneumonia to the mice and it killed the mice, while second strain called non-virulent R bacteria or RUF causes pneumonia but did not kill the mice. I am going to say the answer again because this is a long answer, because there are two strains of bacteria. First strain called virulent bacteria or smooth causes pneumonia to the mice and it killed the mice, while the second strain called non-virulent or RUF bacteria causes pneumonia but did not kill the mice. Next. It's necessary for eggs and sperms or pollen grains contain only half the amount of genetic material found in the other cells of the body. Why it's necessary for eggs and sperms or pollen grains contain only half the amount of genetic material found in the other cells of the body? Because two reproductive cells, I mean sperm and egg, combine to form a zygote that develop to a new individual, carries twice amount of DNA similar to parent individual. Next, scientists thought at first that proteins carries genetic information. Why? You are right, because DNA contains only 4 kinds of nucleotides monomers, whereas proteins contain 20 kinds of amino acids monomers. So proteins can produce more variation of different combination than DNA, as you see. Next, in bacterial transformation, the non-virulent bacteria convert into virulent ones. This is because some of the genetic material transfer from the killed virulent bacteria and enter to non-virulent ones, transforming them to a virulent form. Next, when the mice injected by the virulent bacteria, after killing it by heating mixed with non-virulent strain, the mice died. Good. The answer is going to be because some of the genetic material transfer from the killed virulent bacteria and entered to non-virulent ones, transforming them to virulent, from that causing death of mice. Next, there are only 4 different nucleotides in DNA molecule of living organisms. Why do you think that? Yes, because the sugar and phosphate group in any kind of the nucleotide are the same, while there are 4 kinds of nitrogen base, so there are 4 different kinds of nucleotide. Next, the sugar phosphate backbone is not symmetrical. Good, because it has definite orientation with a free 3' hydroxyl group at one end and free 5' phosphate group at the other end. I am going to say this answer again because it has a definite orientation with a free 3' hydroxyl group at one end and free 5' phosphate group at the other end, as you know. Thank you for good listening. Until we meet again, good luck and goodbye.